Hello, welcome. This is the final lesson in Chapter 1 of Unit A on circulation and immunity, and this lesson is our lesson on the immune system. So, by the end of this lesson, you should be able to describe the main components of blood. Specifically, right now, we're looking at the defense against pathogens in this part of the outcome. And then we're going to analyze the defense mechanisms used by the human body to protect itself from pathogens found in the external environment. This is also all covered on pages 55 to 69, so make sure you review those pages in your textbook. The immune system. So your body is constantly being under threat of diseases trying to get into your body. These pathogens these disease agents are known as pathogens. So there are a lot of different kinds of pathogens. And they can be transmitted by different organisms or vectors. Okay, so an example here of a vector for the disease malaria, the vector is a mosquito. So a mosquito transfers the pathogen, which in this case is a protozoa, Plasmodia, into the human when the mosquito bites them. So mosquito is the vector and the pathogen is a protozoa for malaria. And you can see down below here, there's a bunch of other types of pathogens. We've got viruses. I'll just point out the common ones you should know. Virus and bacteria. We're dealing with those mostly, but you may see like parasites like tapeworms or something like that. But these are the main ones that we are talking about, viruses and bacteria. But there are other pathogens causing diseases in humans as well. So we are lucky, though, we have this physical defense barrier on our body that keeps most of these pathogens out, so that our immune system doesn't have to set up the immune response all the time. So some of these different physical barriers here are listed. These are used to prevent those pathogens from even entering our body. So our skin, we have skin, it keeps everything out, right? Otherwise, everything would go right through our skin, but it's a very strong barrier. We have sweat and oil that keeps things off. Nose hairs that helps catch particles that are going into our airways. The sinuses, tonsils, eyelids, closing the tears, keep things out, eyelashes, hair, earwax, mucous membranes, stomach acid. So if things do go in through your respiratory system or through your mouth, if it's going through your mouth, it hits your stomach, which has a really low acidic pH, which can kill most things. We have normal flora, which is normal bacteria that's hanging out in our guts, and it's really happy there and it's made itself at home. So any other kind of bacteria that wants to make itself home has to outcompete, and that's hard to do. So yeah, there's a lot of different barriers that we have that keep pathogens out. But despite all this, they can still get into our bodies. There's three main access points, and these are cuts or breaks in the skin, breathing them in your respiratory system, or ingesting them in your digestive system. So first thing that happens, if they do come in, you have an inflammatory response, or the second line of defense. So the first line was our physical defenses, and now we have the second line of defense. So if the pathogen gets through that first line, it triggers a series leading to inflammation of where it entered. So wherever that pathogen is nearby there, those capillaries, the white blood cells that are traveling in those capillaries are going to try and squeeze out of the capillaries, and they're going to go, and if it's a bacteria as the pathogen, they're going to go engulf that bacteria. So they kind of like the blob over top of the bacteria. That's called phagocytosis, and brings in that bacteria into the white blood cells, where the white blood cells have enzymes that can kill those bacteria inside them. And you can kind of get some pus here. So there's another diagram of inflammation. It also talks about the capillaries widen here because the blood flow is coming. We're increasing the blood flow to the area where there's been a breach. Because all these white blood cells also want to come to this area. The permeability of the capillaries happens so that the white blood cells can get out and into the cells that have been injured or find that pathogen. So you get some fluid releasing as well at that point. Also, because you're bringing so much blood to that area of infection, you're going to get swelling, pain, heat, fever, because of the extra heat coming with the blood. All right, so you also have an immune response. So this is a specific immune response. So this is very specific to the pathogen that's entered your body. So this response requires antibodies to be formed. So antibodies, there's a term you're definitely going to need to know. Antibodies, they are Y-shaped proteins. There's kind of a picture down here. They're in blue. And they are made specifically by the white blood cells called lymphocytes. All organisms, like bacteria, viruses, other pathogens, even our body cells, every cell has unique markers on the surface of their cell membranes. And these unique markers are called antigens. All right, antigens. So these yellow pieces here are examples of different antigens that might be on the cell surface of a different organisms. So yeah, you'll see that the marker molecules on each of these antigens are different. So this might be an antigen that would be on a virus. This might be an antigen on a certain type of bacteria, etc. This might be a different type of bacteria, etc. So when a foreign antigen, so this is on a pathogen of some sort, like a bacteria, let's say, enters the body, it causes lymphocytes to make antibodies. Okay, so there's the antibodies. The shape of this antibody is going to be dependent on the antigen that's come in. They want to match it, right? So for this antigen, you have an antibody that matches it exactly. It's going to be very specific to that antigen that might be coming in on that bacteria or pathogen. So when the antibody attaches to this antigen, it can help destroy it. Right? They cause it to clump up and make it easier for white blood cells to come in and get rid of them. So here's a little immune response step-by-step process with all the different cells that are involved in this immune response. So first off, your antigen enters the body. So remember, antigens are on the surface of like a virus or a bacteria, some kind of pathogen. All right, so antigens enter. A macrophage, a type of white blood cell, engulfs the antigen and pushes out these antigen markers to the surface. All right, and then we have these helper T cells come along and they help copy this antigen shape so that they can tell the B cells to make antibodies. Okay, so B cells then make antibodies. There's their Y antibodies that match those antigen shapes. These antibodies attach to the antigens. Don't get those two mixed up. Antibodies attach to the antigens, and then a macrophage will engulf them, or the killer T cells come along and puncture the membrane of the bacteria and kills them. So you then, once this is good, everything's going well, the suppressor T cells come along and they can stop the response. And then memory T cells will remember that antigen so that the antibodies are produced faster the next time. Because this whole process takes time, right? It's not immediate. These antibodies aren't just all of a sudden created. These are going to take time to make. So the next time this same antigen enters the body, the memory T cells will be able to pump out those antibodies really quickly. So here's the same process. It's diagrammed a little bit differently, but same idea. You have the antigen, in this case it's a virus, coming in. It's into the macrophage, engulfs it, and puts those virus antigens on its surface, all right, to display them so that the helper T cells can identify them and say, okay, B cells, make these antibodies for us. So you're making all these antibodies, and these antibodies go and attach onto the viruses that are all around the body. Then we have our cytotoxic T cells, which are also known as the killer T cells. So the killer T cells come and they find any of the antigens that are antibodies that are attached to the antigens and destroys them. All right, so here's just a breakdown of the different cells in this process and what their functions are. So we've got macrophages, or phagocytes, engulf foreign invaders, and they present the antigens on their surface. Helper T cells come along, they produce the B cells and killer T cells and maximize the function of the macrophages. They tell the B cells to produce antibodies. Then you have your killer T cells, or cytotoxic cells, and they help kill infected cells. The B cells, they're responsible for the antibody production. Memory B cells and memory T cells keep a record of the infection so they can speed it up for the next time. And then the suppressor T cells suppress and stop the immune response. There's a great video here that you might want to take a peek at. Another person basically explaining the immune response, but it can't hurt to hear it a few different ways at different times. So I suggest listening to that video in your slides. Okay, so we're just gonna look at some autoimmune diseases. So this is a case where the body's immune system forms antibodies against its own tissues. Remember that all our cells have antigens on them, but in this case, we mistake those antigens for foreign pathogens. So if the white blood cells attack our body's own cells, you can get an autoimmune disease. Rheumatoid arthritis is a common example of this, and it's characterized by inflammation in the lining of the joints and stiffness and pain in the joint areas. And the immune response starts with the joints, but then it keeps going for cartilage, bone, tendons, and ligaments. And there's a few other disorders that are just mentioned down here: Crohn's disease, Addison's disease, and diabetes mellitus. So these might be diseases you want to look at when you start looking in the news and listening in the news for some of the different news stories, topics about the content that you're learning in this course. All right, acquired immune deficiency syndrome, AIDS. AIDS and HIV were first identified in 1981, and they think that the virus originally infected monkeys, but then jumped to the human population sometime around the mid-1900s. Most likely in Africa, West Africa, and then the virus soon moved around the world. It has now killed more than 25 million people worldwide. It's caused by the HIV, or human immunodeficiency virus. It looks similar to this rendering up here. And the virus kills and specifically goes after the helper T cells. So this is the problem, because the helper T cells are a really key part of the immune response, as we've seen. Because they're infecting part of your immune system, it leaves these people vulnerable to all the other diseases that might come and attack their body, where the T cells would normally help prevent these diseases. And this virus is spread through exchange of bodily fluids, by sexual contact, or blood-to-blood contact, or contaminated needles. So vaccines. Well, right now vaccines are hugely in the news. Right now I'm making this video in January 2021 during the COVID-19 pandemic. So vaccines are a daily conversation in the news. And our next slide will be about the COVID-19 vaccine. But let's first look at what is a vaccine and how are they created, and what are they used for. So this diagram here, it's from the WHO, World Health Organization's website. I quite like it. So if you have an existing pathogen, something like measles, you probably have the antibody to it already. Actually, you probably have it because you've got the vaccine for it. So you've got these antibodies hanging around your body. Your body knows how to do it. Oh, you've probably had the flu before, so you've got this existing antibody for the flu. You may or may not have chickenpox. I had chickenpox when I was a kid, so I would have antibodies for chickenpox sitting around in my blood. My kids got a vaccine for chickenpox, so they have antibodies sitting around in your blood. But if you have a new pathogen that comes along and you've never had it before and you don't have the vaccine for it, you do not have the antibody for that pathogen that's coming into your body. So what a vaccine does is it lets your body have that antibody and create that antibody before that pathogen comes into your body, so that when it does come in, or if it does come in, your immune system is ready to attack it right away. So how vaccines work is by injecting a dead or synthetic or weak pathogen, right? So it might be a virus or a bacteria, but typically viruses that have been deactivated or dead or like weakened so that they can't cause the actual disease. Okay, and they inject that into the blood so the antibodies can be developed by your immune system. So you're not getting the antibodies. Your antibodies are still being created by your own immune system. You're just getting something that tricks them to think that you are getting the pathogen. So Edward Jenner is given, well, he basically brought this inoculation or vaccination idea to the Western world in 1796. There's a lot of great stories about how this happened. However, a similar technique had been used in India and China many centuries earlier. So it's hard to know if some of that was brought over by Jenner. But anyway, either way, there's a great little story about it. He noticed that so first off, smallpox, a really deadly disease, was going around, killing lots of people. And he noticed that the cow maids that milk the cows weren't susceptible to smallpox. And they would get this other version of cowpox that was a lot less deadly. So that's what he used. What he did is he took some of these poxes, which are like lesions on the skin where I need a picture of that. Look up cowpox lesions and you'll see what I mean. And he took pieces of that where the virus would be and put that into somebody else's blood, right? Made a little cut and inoculated them with the cowpox disease, which cowpox, remember what I said, it's a lot less virulent. It's not going to kill you as much, likely. It's not as deadly as smallpox. And then so the people would make antibodies to cowpox, which I guess is similar enough to smallpox that if you got the smallpox, came into your body, you'd have the antibodies to cowpox, which were close enough to smallpox to be able to fight off smallpox. Anyways, so the idea is inject a dead or weakened pathogen into your blood, and your body builds up the immunity by building antibodies so that you can protect yourself really quickly. An example of vaccine that was very successful was the vaccine for polio. In the early 1900s, polio was a worldwide disease, paralyzing hundreds of thousands of people every year. I can't even imagine. In 1950, vaccines were developed, and in 1980, a worldwide effort began to eradicate polio from the planet. And quite interesting, just recently, this past summer in August 2020, the African continent was certified polio-free. It took quite a while, 40 years. And currently that virus only exists in Pakistan and Afghanistan. If you want to read more, there's a website there. Right, so a vaccine that is totally all over the news right now. You've probably been hearing all about it because it's January 2021 when I'm making this video, is the vaccine for COVID-19. Now, this vaccine is a new type of vaccine. It's been in research stages for quite a few years for other types of diseases, but they decided to use this one for the COVID-19. So this is slightly different than the other way vaccines have been made in the past, in that instead of injecting a a weakened virus or a piece of the virus, they're injecting a part of a code, the RNA code. It's kind of like DNA. It's very similar. Messenger RNA is what it's called. So they take a bit of the messenger RNA code that in that virus would normally code for the spike protein on the surface here. So they take the instructions or the mRNA instructions to make that code, and they inject that as the vaccine. So that code then gets picked up by your cell, and it makes the spike protein that would normally be on the outside of the COVID-19 virus. And they put them up there, and then obviously the body's immune systems will be like, what's that? Comes along and starts making the antibodies like it normally does to destroy those spike proteins, which is great. So the next time your body sees those spike proteins, which is probably if COVID-19 virus gets into your body, they'll be prepared. They'll already have the antibody to attack that virus and prevent you from getting the disease. So be able to attack it right away. So just a slightly different way. It's still dependent on your immune system making the antibodies. It's just what they're injecting into your body as the vaccine is a slightly different thing, right? You're injecting mRNA or the code to make a piece of that virus. Okay, sometimes these can get confusing, confusion between the two, so make sure you don't mix them up. What is an antibiotic? An antibiotic is a chemical that kills bacteria. So usually you take it by pill if you've got a bacterial infection. It usually affects how bacteria makes proteins or prevents them from building their protective coat on the outside of their cells. So it only works on bacteria, and you ingest it. What is a vaccine? Well, as you've just learned, a vaccine is like a weak or dead pathogen that you inject into your body to create and trick your body into making an immune response so that you have antibodies to fight off future attacks. All right, so, and vaccines are for viruses. You take it once, it's got permanent effects. Usually, you might have to do some boosters later on. So it's a preventative thing before you actually get the virus. Whereas antibiotics, you don't take until you actually have the bacteria in your body, and you take it after you've gotten infected. So one thing you might see in the news is what is antibiotic resistance. And this is because this resistance can occur because bacteria's DNA can mutate and change really quickly. And some mutations can cause the antibiotics to not work anymore on the bacteria. And these resistant bacteria survive, and then they continue to infect and replicate and grow. And now you have an antibiotic resistant form of the bacteria. All right, these are some of the resources used to create this presentation. That's the end of the lesson. So make sure you review your textbook, you do your practice questions, and your self-check questions too.